thanks everyone for coming. Can you hear me okay? Yeah? Okay. So I'm a postdoc at the University of Queensland and the Australian Institute for Marine Science, and I'm excited to tell you about our new coral work. So these results are very, very new, so if you do have any insight, or if you've seen similar patterns in other organisms, please do come chat to me after. So genetic biodiversity is not always packaged into tidy species groups, and instead, as population geneticists, what we more often see are these confusing patterns in natural populations, where individuals fall along a continuum of genetic diversity, of genetic differentiation, and it's actually hard to decipher which groups of individuals form discrete interbreeding entities. But understanding this process and how and when species diverge from each other and whether they experience independent histories of adaptation is a central question in evolutionary biology. In the marine environment, delineating such cryptic diversity can be complex, so corals form the basis of reef ecosystems which harbor exceptional biodiversity, but they're different from terrestrial systems in several ways. So first, most species divergences predate modern-day reef configurations, which are only about 12,000 years old. So the species distributions that we see and the genetic diversity that we see is really a product of repeated glacial cycles, recurrent sea level changes, and messy recurrent secondary contacts that have occurred over very recent evolutionary timescales. So as a result, marine environments often host closely related sibling species that are often found in very close sympatry. And because larvae can spend weeks in the water column, most sibling species can sustain very large overlapping populations over spatially complex habitats. And in these habitats, they have lots of opportunities for interspecific gene flow. And with this process, it might also enable these sympatric populations to co-adapt to changing environmental conditions. So from studies in other organisms, it's well established that hybridization can be an important source of adaptive diversity, either by boosting or reshuffling existing genetic diversity into new allelic combinations. Selection on parental ancestries can also result in adaptive introgression, where selection for specific alleles can sort of get them preferentially transferred between hybridizing taxa, while the rest of the genome remains highly differentiated. It's also evident that local environments can directly affect the strength of reproductive barriers, such that the repeatability of species boundaries and the outcomes of hybridization across species ranges can be highly variable. So in corals, hybridization has been suspected for a long time, and now with genomic data, we know that gene flow is very common and that it can be highly heterogeneous across the genome, but most of these inferences come from demographic modeling. We also have limited evidence that introgression can be adaptive and involve the influx of genes from closely related species. And in particular, these are 4 cryptic species of Acropora hyacinthus that are found in American Samoa, and these species show marked variation in bleaching susceptibility. And Rose et al. Found that the cryptic taxon HE, which also bleached less, has sort of a large chromosomal chunk that shares high genetic similarity with the closely related Acropora millipora. So the authors suggest that it could have been acquired through the process of introgression. And because this locus also showed high rates of protein coding substitutions, the authors posit that perhaps introgression can be involved in adaptive diversity and also a diversity that's shaping thermal tolerance. However, we still know very little about how genetic differences are maintained between closely related coral species and whether introgression can actually facilitate adaptations across species ranges. So in this study, we sampled over 900 corals across the expanse of Australia's Great Barrier Reef to examine the role of introgression in the evolution of the widely distributed Acropora hyacinthus. So our specific aims were to resolve whether hyacinthus in the Great Barrier Reef comprises multiple interbreeding taxa and whether introgression, we wanted to investigate whether introgression has played a significant role in shaping adaptive diversity across the species range. So we used low coverage whole genome sequencing of populations from 26 reefs, so including extremely remote environments in the south and the far north of Queensland. So you can imagine this was a massive, massive effort involving lots of researchers over a 4-year period. So once we had the sequences, we aligned them to the reference genome, and then we identified polymorphic sites and estimated genotype likelihoods using ANCs, and then conducted a series of population genetic analyses for genome-wide introgression and also locus-specific introgression. Okay, so here are the results. Sampling across the Great Barrier Reef, we found 4 genetically distinct taxa, which are shown here in the PCA. So all of these samples were collected as Acropora hyacinthus in the field, suggesting that these species, well, these taxa are truly morphologically cryptic, at least under field conditions. And if we look at this admixture analysis, which is flipped on its side with individuals ordered by latitude, so you can see that all 4 colors corresponding to the different clusters actually occur broadly across the Great Barrier Reef, and that they also occur sympatrically at many of the reefs that we sampled. So in fact, at many of the reefs, they actually coexist in the very same environments, but genetic differences are maintained despite external fertilization and lots of opportunities for interspecific gene flow. We also found that our 4 groups show genetic affinities to Acropora hyacinthus sampled in American Samoa. So shown here, I don't have a laser pointer, I don't know why I'm doing that. Sort of segregating on the first PC axis there, suggesting that their ranges might actually expand far beyond the Great Barrier Reef. And when we compared all these species to closely related Acropora mellipora, we could confirm that all of these hyacinthus taxa, so for both the GBR and also American Samoa, are indeed more genetically similar to each other, and that they likely form a closely related species complex. So in light of limited evidence for adaptive introgression between mellipora and taxon HE in American Samoa, we first tested for introgression between mellipora and our Acropora hyacinthus clusters in the Great Barrier Reef. So we did this using genome-wide D statistics and Acropora tenuis as an outgroup, and then we also examined these signatures within each region of the GBR separately to understand better how these patterns vary across the species range. So we found significant introgression from mellipora into groups 2, 3, and 4, and these signals were consistent across all the regions of the Great Barrier Reef. However, when we examine the fraction of admixture, so FD allows you to identify parts of the genome that show high proportions of shared derived ancestries between introgressing species. In general, there were only few regions that showed high FD values that could be indicative of adaptive introgression. And just looking at these 2 chromosomes as an example, you can see that these FD peaks are sort of scattered across different chromosomes, and we found no compelling evidence of large chromosomal regions that are involved in introgression between these species. And because these signals were consistent across all 3 hyacinthus taxa, we could conclude that, well, introgression with mellipora has likely been historical and predates the divergence of at least these 3 taxa, but we are going to explore this with demographic modeling as well. Okay, so now focusing only on the hyacinthus clusters, we find complex patterns of population structure with latitude in these 3 genetic groups. So here, south is blue. And looking at the individual PCAs of each group, we can see that some populations in the southern part of the range actually show distinct genetic segregation from populations in the north and central regions. And furthermore, when we looked at admixture proportions, we found that these distinct southern admixed groups are comprised, well, I gave it away, are entirely comprised of admixed individuals, whereas early generation hybrids are only found in the northern and central parts of the range. Okay, so what is going on in the south? Well, you can imagine in this hypothetical data set, we can define a triangle between 3 different parental taxa that are shown here in a hypothetical PCA. So under 2-way admixture, we would expect that admixed populations would fall on the line of the triangle between 2 hybridizing taxa. However, under a 3-way admixture scenario, admixed populations are going to be pulled inside the triangle because they share genetic ancestry with all 3 parental populations. And indeed, in our own data, this is exactly the pattern that we see. So southern populations are being pulled inside the triangle, suggesting a 3-way admixture in the southern part of the range. So this pattern was also supported with significant migration edges between the southern admixed populations in a tree mix analysis, and also these statistics indicated three-way admixture, but only in the southern range, with no significant evidence of gene flow in the central and northern regions. So this suggests that reproductive isolation is likely variable across this small species range, and that selection on hybrids could also be variable and depend on the ecological context. So now we have a complex system of highly localized admixture between three divergent lineages at the very marginal edge of this distribution. So at these southern reefs, non-admixed and admixed individuals actually coexist in the same habitats, implicating some level of reproductive isolation between them, and possibly selection on admixed genotypes if parental populations are maladapted at this southern range edge. In fact, we found that admixed populations deviate from parental non-admixed populations along the same axis of variation in a PCA, which would not be expected under neutral evolution. So to investigate whether admixture has resulted in local adaptation, we computed allele frequency differentials for three-way admixture. So here the change in allele frequencies is calculated between admixed and non-admixed populations. And because AFD3 is simply the product of the three allele frequency differences, we would only expect very high values for loci where all three populations have experienced convergent allele frequency changes away from their parental populations. So plotting these allele frequency differentials, you can see that high values are distributed all sort of across all different chromosomes. So rather than selection acting on large chromosomal regions, which we were expecting, parallel divergence appears to be highly polygenic, and it's localized to multiple in small chromosomal regions. And just to give you an example, we see several islands of parallel divergence with high FD3 values, where all three admixed populations show these concordant deviations in FST from non-admixed groups, suggesting that selection is acting on the same regions independently in these admixed populations. And because there was a lack of fixed variance within these regions and a general trend toward high diversity around those regions as well, selection has likely been recent and is acting on admixed variation, but we have yet to test this formally. So just to conclude, we find four sympatric taxa that are co-occurring across the Great Barrier Reef. Introgression with Mellopora has likely been historical and predates the divergence of at least three of the hyacinthus species. We find localized three-way admixture at the very southern range edge, suggesting variable reproductive isolation across the species range, and possibly selection for admixed genotypes, but only in certain ecological contexts. And this is really exciting to find because we know so little about the speciation process in these high gene flow systems. So consistently, we find evidence of parallel differentiation following admixture, suggesting that admixture has somehow unlocked convergent adaptation, either through selection on alleles that have been acquired through introgression, or possibly those originating independently in the admixed populations. So this suggests that introgression, or natural assisted gene flow can occur, and that we don't know yet if it's fully adaptive, but that this process is occurring, and it can perhaps enable populations to co-adapt to variable environmental conditions or to overcome sort of complex drift dynamics that might be occurring at their range edge. So thank you so much to all the people involved. It's been a huge effort, and thank you for coming.
Okay, thank you for your patience, everybody, in more ways than one. This is it. I think it's the final talk pretty much. So I'm going to be talking about this question: does selection favor the maintenance of porous species boundaries? That work? Yes. Classical theory of species and speciation was originally based on this idea of complete reproductive isolation, and that as a result of that, species evolve independently. So here, this is kind of representing two species over a number of generations, and gene combinations are getting shuffled up within each of those species. But there's no exchange of alleles between them. So why should it be like that? And there's lots of explanations, of course, but one way to think about it is a model of adaptation to divergent ecological niches. These are classical models by Felsenstein and a version by Dial and Bush as well. So here I'm representing there's two ecological patches that these palm trees are living in. And so there's divergent selection between those. There's a different optimum in each patch, but there's some amount of gene flow between them. And so the classic result there is the amount of divergence you get will be, it will increase as the strength of divergent selection increases, but it will decrease as the amount of gene flow increases. And although you can get divergence in the presence of gene flow, the optimum fitness across the whole system would derive from having no gene flow at all. So if there's any traits that selection could act upon to reduce that level of gene flow, then potentially that would happen. So you get selection for reinforcement. But at the same time, we've heard today and across the 4 days, of course, lots of examples with evidence for gene flow between species. I'm just showing some graphically here from a review by Endelman and Mallet in 2021. So there's integration happening between mosquito species and among sunflower species and so on. So we've got this kind of alternative scenario where now we've got our two species over time, but there are actually alleles that can cross between these species. So how might we kind of reconcile that, or what hypothesis might there be for this observation of what I'm going to refer to as porous species boundaries? So one possibility is that these are kind of steps along the way to complete isolation, so they're kind of partial cases of partial speciation, if you like. It could be that there could be issues with the methods for estimating gene flow, and maybe we're overestimating that, and there's been a lot of work on that over the last few years. It could be that selection is in fact favoring the closure of these boundaries, so like those models of reinforcement, but there are constraints somehow. So perhaps selection, once gene flow is low enough, then selection just becomes vanishingly weak, so it's never going to complete closure of the boundaries, or there might be some kind of mutation-selection balance, so you never get to a completely closed boundary. But the hypothesis that I'm going to think about today is that selection might actually be favoring the maintenance of porous species boundaries. So we'll think a bit about how that might work. And to start with, I'm going to do that by revisiting these sort of classic models of adaptation in distinct ecological niches. So we've got our two patches again, with palm trees in the two patches. And we've set up, so it's a haploid model to keep things simple. There's two loci, A and B, and little a, little b is the optimum genotype in one patch, and big A, big B is the optimum in the other. And then we've got a way for there to be assortative mating, so to reduce the amount of recombination between those optimum genotypes in each patch. And to keep things relatively simple, we're assuming that's a pleiotropic effect of the resource genotype. So there's actually two mating pools, and the probability of entering the early mating pool versus the late mating pool is different for the little a, little b, and the big A, big B genotypes. So the patch specialists are tending to head into an early and late mating pool, whereas mixed genotypes are just entering those pools at random. So then there's two additions to the model compared to the sort of classical version. And one of them is in addition to, so the two patches have these aspects of their environment that you could think of resources that are there, so that's what's being determined by the A and B loci. But now we're thinking about some additional part of the environment, and I'm going to refer to that as the physical environment. where, again, there's two environmental states, but these can vary over time. So these can vary independently of the differences between the two patches in terms of resources that are present there. And so at any point in time, the physical environment can be the same in both patches, or it could be different between the two patches. And those are going to be determined by two additional loci, so C and D. And then finally, in order to see how selection might act on the amount of gene flow that occurs, then I'm considering another locus that's a modifier for the strength of assortative mating. So whether the optimum genotypes for the resource aspect of the two patches, whether they mate assortatively or not, depends on this other locus. And there's just big E means that they mate assortatively, as I've already described, but if that mutates to little e, then that means they mate at random as well. So the frequency of this little e locus will determine to what extent the whole population, their mating is either assortative for the specialists in the two patches, or whether it occurs at random. Okay, so first of all, if the physical environment is just constant and different between the two patches, and that's just adding extra strength of divergent selection in addition to the resource optima in the two patches. And in this model, I've started a run here. So along the x-axis is time in generations. And this panel up here is showing the frequency of assortative mating. I started with 100% assortative mating, and it's declined to a level that's determined by mutation selection balance in this case. There's a certain number of these random mating loci are just appearing through mutation, but they're being selected against, determined by the selection coefficient against hybrid genotypes. But what happens then if there's a different pattern? So here we've got, the physical environment now is actually fluctuating over time, and it's fluctuating in a way that is out of phase. So at the start, patch 1 has the grey physical environment, patch 2 has the white physical environment, and then they swap values every now and again. And so what this means is that immediately after the shift, then the optimum genotype in each patch is requiring alleles that were previously in the different patches. So you have, in patch 1, little a, little b is always favored, but you're fluctuating between whether little c, little d, or capital D, c, capital D are favored. And so recombination between the two patches is favored at those points when the environment swaps between them. And so what you can see then is that so this panel is showing the frequency of assortative mating, and so there's pulses after each shift in the environment where that drops, and this is because the random mating allele is spreading. So that's spreading through its association with the favorable genotypes that are created by hybrid matings, if you like. So of course it's unlikely the environment would shift like that between two patches, but if you have random shifts in the environment, then you can get the same kind of thing. When the environment shifts in a way that favors the transfer of an allele between the two species, then you get the spread of random mating. Okay, so that's a fairly simple model, but it's kind of highlighting what sort of conditions might actually favor porous species boundaries. And it's the idea is that if there's a shifting environment, then recurrently you create the conditions where it's actually, there's a tendency for some alleles that are in the other species to be beneficial to receive in the first species. So these are the conditions that might favor. There's, first of all, you need there to be non-zero gene flow. You need the condition that imported genes actually bring net or recurrent benefits. So it's not just a one-off case of a contact zone where some introgression occurs, but then that stops. But there needs to be some reason why there's constantly this tendency that some alleles from the other species are going to be beneficial in the first one. you would need that there's genetic variation in the mechanisms that actually affect partial gene flow. And as I've already emphasized there, these conditions would somehow need to be maintained over time. And if all of those things happen, then potentially there could be selection that's affecting the level of gene flow and actually favoring a non-zero level. In this recent article, I looked through the literature to kind of tot up evidence for those different conditions. And I don't have time to go through all of this now, but I think the key point is that a lot of the conditions are kind of met in terms of there is non-zero gene flow. We can see cases of adaptive integration where there are clear benefits of receiving alleles from another species. But the key question is whether those conditions could be maintained in a net or recurrent way that would kind of maintain that selection rather than just being a one-off event. And in terms of whether there's evidence that selection does affect the level of gene flow, yes, there is in terms of evidence for reinforcement. But in terms of evidence that it might sometimes favor the increase in the amount of gene flow, then there's a handful of cases of those have been presented. All right. The same kind of ideas could be applied to bacteria, and maybe between bacterial species, the starting point would be the expectation that it is kind of beneficial for alleles to be, or genes to be shared and swapped between different species because of frequent observation of horizontal transfer. But the same kind of mechanisms would apply. So what are the specific conditions where it's actually beneficial to receive alleles from those other species? Okay, so to conclude, porous species boundaries could actually be a transient step on the way to complete closure, or they could be kind of an incidental outcome of how adaptation is occurring in each of those species. But there are these conditions and scenarios where selection might favor porous species boundaries. And why might this be important? Well, this is really going to determine how species collectively adapt to environmental change. So are they doing that independently, or are they doing it in a parallel way? Independently and in parallel, or are they doing it by sharing beneficial alleles? So in terms of opportunities for future research with this, one thing that would be very useful, and indeed there was a talk in the previous session that had done this by Xavier, is systematic surveys of gene flow. And the opportunities for this will increase with emerging genome data to see what proportion of species are actually displaying porous boundaries. And perhaps existing studies have been a bit focused towards those because of the interest in understanding those systems. It would be necessary to try and estimate the selective gradient on gene flow if we're thinking of that as a kind of trait that might evolve through these kinds of mechanisms. And then finally, it would be very useful to perform experimental evolution where you could actually modify the environmental pattern and patterns of fluctuating selection, for example, and see whether you can observe changes in levels of gene flow evolving in those cases. Okay, thank you.
Thank you. I think we can take a question or two because we're near the end of everything. So we don't need to keep back about time. Do you know of any cases where people have tried to do the second, estimate selection gradients on gene flow?
Yes, the question was whether anyone has actually tried the second approach of estimating selection gradient on gene flow. There is, I should show, so what I was talking about here is in this article at the top. So in there is an example that was a bacterial study where they did actually measure the fitness effects of transferred bits of DNA. And so there is one one case like that I know. Yeah. Yes?
Might whether selection favors a porous or a closed boundary vary across the genome?
Yes.
So if I'm sitting next to a diagnostic-molar incompatibility, I probably don't want selection, but if I'm a mitochondria, I might like to introgress like all hell.
Yes. So, I mean, one sort of mechanism that might arise if selection was acting in this way is that the genome could partition in a way that some parts maintain high gene flow, but other parts are protected. Yeah. We should probably stop. I'm happy to talk to anyone. Thank you very much.
Okay, thank you everyone for persisting until the end, and special thanks of course to all the speakers of this symposium. You've made this a very interesting and a diverse day. In this last slot, I attempt to draw the themes of this symposium together, highlighting some outstanding questions from my perspective, and then leaving some time at the end for a joint discussion. But before I do that, I wanted to mention an activity that might be of interest to you. And this is the SF-funded special topic network called Integration of Speciation Research. And the aims of this network are to promote a framework for integrative speciation research and tools for comparative analysis, but also to promote integration of speciation researchers, diversity and inclusiveness in our field. And in a recent perspective paper that resulted from a workshop that we organized, we detail how our community could benefit from sort of further integration and what could be some of the practical steps towards that. And in addition to these workshops, we also organize a seminar series that runs monthly, and all our seminars can be found online, recorded, so you can watch them later as well. And something that has been occupying our minds lately is setting up a speciation database that we hope to be a comprehensive database that can include all taxa, all reproductive barriers, and in the end become a resource for the community that people can use for comparative studies. But then back to the topic of today. So one way to define speciation is that it's the evolution of reproductive isolation through the accumulation of barriers to gene exchange. And I wanted to mention that in the past years, there has been conceptual work done highlighting that some of the common terms we use might mean slightly different things to different people, or that we might use different terms to mean similar things. And that's okay. We might not be able to agree on all definitions as a field, but then it becomes important to define what you mean by the key terms you use. So I'll try to do that here. So barriers are genetically based obstacles to gene flow, and they can come in many flavors. Or alternatively, one could think about the components of reproductive isolation. So strength of habitat preference or strength of hybrid inviability. And the theme that we wanted to draw together in this symposium was that these barriers or components of reproductive isolation are often studied in isolation of one another, but their interaction or coupling may change the dynamics of the speciation process, and it could speed that process, or it could also halt or reverse that process. Another theme that has been floating around a couple of years in our community is this question of persistence. So do these emergent lineages persist until speciation is completed? And do species persist over macroevolutionary timescales? And what determines the persistence at these different timescales? So it may in fact be that the initiation of divergence is easy, but persistence of these lineages might be difficult. So this symposium draw attention to 3 questions that we think are outstanding. And one of them was, how do barriers interact and couple during speciation? A second one, which barriers are important for coexistence of lineages and what determines persistence? And finally, how do processes at microevolutionary scale connect to macroevolutionary patterns and processes? And I'll very briefly go through these 3 questions, picking up some of the themes that were discussed in the talks today. So how do barriers interact and evolve during speciation? So we have sort of examples from different types of interaction or joint action. So one barrier can promote the evolution of another barrier in some cases, whereas in some other cases it seems that the additional barrier sort of gives diminishing returns in terms of increasing the reproductive isolation. We also heard several talks on the interaction or coupling between barriers at the genomic level. And we heard about chromosomal fusions mediating genomic coupling, and also multilocus interactions mediating genomic coupling. And one theme that I think about a lot is the interaction between ecology and incompatibilities that maybe wasn't touched upon today in this sense. So under some scenarios, ecological divergence can allow accumulation of truly intrinsic incompatibilities. But then in other conditions, actually the interaction between ecology and incompatibilities hinders the evolution of strong reproductive isolation, and this is the scenario where the incompatibilities actually depend on the environment, so that hybrids have a low fitness in the parental environment, but if the environment changes, actually the fitness of hybrids and parents could be on par each other. The second question was, which barriers are important for coexistence of lineages and what determines persistence? Some themes from this symposium were ecological differentiation is important for coexistence. And just now we heard that some gene flow may actually be important for persistence, so sort of a counterintuitive claim, because this gene flow might allow adaptation if the environment changes. So putting that together, coexistence and persistence do not actually necessarily require completion of reproductive isolation. And finally, to the third theme, how do processes at microevolutionary scale connect to macroevolutionary patterns and processes? And themes from this symposium, we heard about caves, that there's rapid sort of population formation, but there's also a higher chance of extinction, and if I understood it correctly from the talk, then this is actually decoupled from the rate of speciation completion. And interestingly, we also heard that plants evolve reproductive isolation faster than animals, even though they also seem to experience more gene flow. And inspired by the plenary talk on the first day, something that I started to think about was what are the units of comparison, or do we know what the units are actually? So units of comparison, they might be different at the microevolutionary level compared to macroevolutionary level, and this might make the comparison between two scales difficult. But now we actually wanted to turn the question to you and ask, what do you think are the outstanding or interesting questions that are related to these later stages of speciation from your perspective?
The floor is open. Comments, questions, however stupid or off the wall they might seem to be. I've got a stupid question then. Is persistence thing part of the speciation process, or is that something else?
I don't know. I guess I maybe it depends. Are we talking about persistence of these sort of emergent lineages? Do they persist until they become complete species? Or do we talk about persistence of species after speciation has been completed? So do these species persist over macroevolutionary timescales?
So I'm not really proposing any outstanding questions in this field, but rather raise a question about how do you define the late stage of speciation? Because I just want to use an example of my own research that when we study reproductive isolation between like two species where we define them as species, but there's some like there's insympatry, and we got hacked by saying that, okay, whatever reproductive isolation you found between species, you don't know whether it's important for the speciation, right? Because it could be accumulated after the speciation completed. So, but when you say at the later stage of speciation, exactly what are you talking about then?
I'll hand it over to Roger. You can answer this.
Well, I mean, I think there are two somewhat different ways of thinking about that, which have been referred to during the meeting. I mean, some people would say we're getting to a late stage when it becomes possible for ranges to overlap, and others would say we're getting to a late stage when reproductive isolation is nearing completion or gene flow is nearing zero. And those two things may go together or they may not. I think it was part of Sean's point earlier, for example.
Yeah,
it's an important realization that they're not necessarily going together, those two things.
Yeah, because I would just feel like the field don't have a consensus of how you define later stage of speciation. At least that's my experience by talking to different fields. Yeah.
So yeah, I actually had the same question. And I think, again, we sort of view these kinds of questions within the lens of our empirical or theoretical, constructs. And I sort of feel like the corn borer is not late stage speciation, right? But I think that what I'm interested in is the degree to which independent phenotypes are becoming associated with one another, each of which contributes to some amount of gene flow reduction. So for me, the way I think about, I'm not sure it's late stage speciation, but sort of like the accumulation of independent phenotypes, each of which contributes to partial reproductive isolation, and the dynamics under which they become associated or it reverses and then become decoupled. So I think that the phrase late stage speciation is sort of applying a very concrete idea on what is definitely a continuous process. And so maybe this is a situation in which we can expand our language a little bit to better accommodate that continuum.
And maybe just to comment on persistence in that same context and thinking about our own study organisms. So if you think, I think a lot about the crab and wave ecotypes of Littorina. I wouldn't describe them as a late stage of speciation because they're parapatric rather than sympatric. They can't coexist. There's a high level of gene flow between them. But I think there's good reason to think that they're persistent. They may persist for a very long time in their current state of incomplete reproductive isolation, and they are distinct in many phenotypic traits that are held together because of adaptation to the same environmental axis. But that's a different sort of persistence from the persistence of coexisting in reproductively isolated groups. And so again, as always, language is getting in our way.
I think maybe one possible way to think about it, I tend to think of it this way, is it's a late stage
some other means than hybridization. So they might not persist very long, but the way that they go extinct is not just going to be they collapse back into a hybrid swarm.
Yeah, but the thing is, how do you know whether it will go extinct or
Yeah.
not? Like, we're having just a snapshot.
Well, I mean,
of-
everything's going to go extinct.
Yeah, that's true. So that's like, again, back to this kind of murky definition of later stage of speciation studies.
Anyone else?
Any other comments? Thoughts?
A slightly provocative question, maybe. A lot of the talks incorporated some level of gene flow, either in the models or in the empirical work. And I guess I'm wondering whether we should be thinking of gene flow as a null rather than allopatry as a null. And how many allopatric things are there? And how does RI build up in allopatric things, or allopatric species pairs? So that's...
Does anyone have a thought on this? Well, I've seen many references to Sean, your work on allopatric speciation lately, so I don't know if you want to comment on this.
Just how things build up in allopatry?
Well, I guess I'm wondering whether we should be studying allopatric pairs rather than the inverse, thinking about sympatric with parapatric kind of exception. Maybe that's an outdated view. I don't know.
Yeah, I agree 100% that we should be studying allopatric pairs a lot. And I loved your paper. I thought it was really amazing. Because of the definition being the evolution of reproductive isolation, I think it's natural that everyone's looked at for the last long time things that are kind of not fully there, or things where you can test it. So reproductive isolation obviously is tested where they meet. But if the things that are important to it happen where they don't meet, then yeah, obviously it would be really helpful to get more information on more allopatric evolution, I think, for sure.
Any final thoughts?
We are anyway well past the time, and I know everyone's thinking about the dinner this evening. So thank you all once again for coming, and I think it was a great afternoon. Well, great day, actually, whole day. And we look forward to seeing everyone later at the social. Thank you to all the contributors.